welcome to our functional MRI speaker series for April. We have a very special guest today, Dr. Thomas Liu from the University of California, San Diego. Tom got his BS at MIT in electrical engineering, and then continued to study more electrical engineering at Stanford University, where he got his master's and his PhD. Then took a short break to go to industry, where he worked for Acusun, doing signal processing for ultrasound applications, and then went on to do his PhD at Stanford and his master's, processing data on magnetic fields of the Earth for trying to detect earthquakes. Really cool stuff. After that, he went on to do his postdoc at UCSD, where he stayed, got a faculty position, and he is now a full tenure professor, as well as the director of the fMRI Center over there. He's done a lot of really exciting work with the Bold effect and with ASL, and a lot of it trying to understand the signal properties and cleaning up the noise. And recently he's been doing a lot of work on looking at the global signal and regression and what it means, and how the different signals relate. And he's also an editor for NeuroImage. It was. Oh, you stepped down.
Yeah.
Congratulations.
Yes.
Anyway. Yeah, exactly. So I used to get a lot of messages from him about, like, where's my article? How's that review coming? Anyway, on top of that, he's a really nice guy. We've had a lot of very productive discussions since he's been here. And without any further ado, I'll turn it over to you, Tom.
Great. Thanks, Luis. So thank you all for coming. So today I'd like to talk about 3 different topics in resting state fMRI that we've been looking over the past, say, 5 years or so. So the 3 things that we'll talk about are the global signal, vigilance fluctuations, and nuisance regression. Now, actually all 3 are related, so actually I'll be talking about all 3 at the same time in a way, but we'll try to, that's the general scheme of things. So I think most of you are familiar with resting state fMRI, and so I'm not going to spend too much time on this slide. And the basic idea with resting state fMRI that you all know is that the resting state functional connectivity maps that you get here in the resting state are very similar to the task-related maps you would get if you actually tapped your finger. So this was first observed by Vigilance and others at Medical College of Wisconsin. And since there's a lot of students in this room, I always like to tell the story to students in the fact that, according to Vigilance, this paper was rejected at least 3 times because no one believed it was true, that it had to be an artifact. And I think finally Jim Hyde, who's a big name in the field, only because he hassled the editor so much was it ever published. And so I think that's a lesson that when sometimes something is too new, it's rejected. And then once it's, after a while, it's considered to be obvious. Okay, I think there's a quote from Schopenhauer, is it? That at first it's violently rejected, and then later on it's like, well, that was really obvious. So resting state fMRI is like that, and now the NIH has been spending hundreds of millions of dollars to do this method. So in today's talk, I'm going to talk about 2 different types of measurements that you might do from resting state fMRI. One is the static functional connectivity estimates, where the simplest example is if you have 2 time series, you might simply compute the correlation over that period, so over the entire scan, and so you have a single functional connectivity value for that entire scan. So the first part of the talk will mostly be about static functional connectivity. Then we will also talk a little bit about dynamic functional connectivity, where it's identical to static functional connectivity, except now you have a sliding window and you're looking at the correlations over time. And typically these windows are on the order of, say, 20 to 40 seconds. And so you're getting you're looking at these correlation within a smaller time window. We'll also be talking quite a bit about modulation of functional connectivity and what are the factors that might affect it. And our first study in this area was done back, I guess, 2009. It was published, which was we looked at functional connectivity during rest. We also did some finger tapping to to verify where the networks were. And then what we did was we gave people 200 milligrams of caffeine and looked at the conductivity afterwards. And what we found was that caffeine totally wiped out or significantly reduced the conductivity in the subject population we looked at. And so at the time, we didn't really understand what was going on, and so a lot of this talk is going to be sort of the history of what we did afterwards to try to understand what might have led to this decrease in conductivity. But I think an interesting study that came out around the same time, also from UCSC but totally unrelated, was this study where they, by Sarah Mednick, who is known as Dr. Nap. If you Google her, she does lots of nap research. She's now at UC Irvine. And so she did the study of learning with either placebo, nap, or caffeine. And this was very interesting in the sense that this was a motor learning task. And so if subjects learned the task in the morning and then either did their normal routine or took a nap or took caffeine in the intervening 4 to 6 hours, came back and were tested again, then what they found was, I believe nap and placebo were pretty much similar, but as you can see, there's a really big decrease in the motor learning with caffeine. Okay? Now, this didn't apply for other types of learning, such as verbal learning, but for motor learning, it seems like whatever, this reduction in conductivity might have been related to this reduction in motor learning. So the takeaway message for this is if you're trying to learn a new piano piece, don't take caffeine, okay, or anything to do with motor learning. Other things like cramming for a test, it's still probably okay. All right. So a lot of this talk, we're going to, most of you have done fMRI analysis, and so I think everyone in this room, or many of you, have done what's called nuisance regression. It's a fairly standard method for fMRI, and the idea is that the data we measure, so for example, this time course, we believe there's things that are confounds of no interest. And so for example, in this case, the experimenter has measured different motion parameters, and then the idea is to take these motion parameters, regress them out of the signal measured, and then you end up with your cleanup signal. Okay? So I would say probably 99% of fMRI studies do some type of parameter regression. So certainly motion regressors are commonly used. Other regressors that might be used are things like heart rate or heart rate variability. Respiratory activity is used quite a bit. And then here are some motion regressors. And there is this thing called the global signal, which is shown here in black, which we're going to spend quite a bit more time talking about. Now, the global signal is, one way to think about it is this is sort of a way of, this is called what Jonathan Power calls a gray plot, which is essentially, this is space, so these are voxels, voxel dimension, and time, and so it's basically taking all your data and representing as a 2D matrix. And as you can see, basically, as you're going along time, there are these very strong spatially widespread artifacts that occur, okay? And if you imagine you're just averaging along each column, then what you end up with is the global signal. All right, so it's really simply the mean signal that you obtain, all right? Some other fun ways you can get the global signal are here in, let's see, in blue is the global signal. And then in green, this is from the group at McLean, is measuring oxygenation at the fingertip. You can actually get something that's related to global signal. They've also done stuff where you actually, if you measure oxidation at your toe, you can actually get something that's highly correlated with this global signal. And here we're showing some data measuring sort of from sagittal sinus or the great vein of Galen, which sort of drains down here. And so there does seem to be, the fact that you could measure it sort of at these sort of distal areas seemed to sort of suggest that it was a good thing to regress out, that it really seemed to be a confound that you should be taking out of your data. And so the process of global signal regression is fairly straightforward. You compute your global signal and you just regress it out of every time series, and it's done independently on every time series in your data. Okay? So when I first heard about this, I thought this was a really stupid method. The grad student who was visiting at the time wanted us to look at it, and I thought that's really stupid. Why would you ever do that? You're using the data twice. You know, it's sort of just going against everything you should do, but he convinced me we should look into it. And so 10 years later, we're still looking at it, and he's doing something else now. He's still doing fMRI, but just not global signal. So here's the main reason why you want to do the global signal, which is these are resting state correlation maps from 5 different subjects. And if you look at the top row, you can sort of see you have something interesting for subject A, but B, C, D, and E are, if this is your data, you're not going to publish your paper, right? There's just nothing that's too interesting there. And so if you apply global signal regression, all of a sudden you get these really nice maps, at least in these 4 subjects, not so much in E, but you're starting to see what's called the default mode network, where the red is the default mode network, and the blue areas here are sort of your task positive or your attention networks, okay? And so those 2 networks have, there was actually quite a bit of excitement over those 2 networks when the first paper, Fox and Rakel, published in 2005, where this was just really sort of intriguing idea that the brain is sort of always going between these 2 networks. So here in orange, you sort of see the default mode network, and when it's going up, this other network is going down, and when it's going down, the other network is going up. And so this dynamic picture of the default mode network as sort of your sense of yourself, your self-rumination, your interior world, and then switching to this more exterior-focusing view of the world, and that it had a nice subjective correspondence with what we sort of experienced, that maybe that maybe is how the brain works, that there is this anti-correlation between these 2 networks. Okay, so I think actually this paper contributed to really sort of why resting state fMRI is now still being used 14 years later as one of these pivotal papers that got people really excited about this area. Okay, because you realize that this paper was published 10 years after the original paper by Biswal, and it really took 10 years for people to start accepting this and to get excited about it. And now, 14 years after that paper, it's now considered standard. Okay. Now, these data were obtained with global signal regression, and later on, Kevin Murphy from the NIH pointed out that, well, that's all fine and good, so you can obtain these nice anti-correlated networks if you do global signal regression. But if you don't do global signal regression, you don't really see these anti-correlated networks. All right? And so the question was, are these anti-correlated networks simply an artifact of your processing? And one could argue, this is a very simple picture, but if you imagine, these 2 vectors here represent time series from 2 different voxels, and let's say all your time series are spread around this cone, then the mean of all those time series is simply this red arrow in the middle. If you regress it out of these 2 guys, then they point along different directions, and now they're anti-correlated. So they're correlated, now they're anti-correlated, and it just has to be. And so the argument was that even though in the original data there's no anti-correlations, by doing this process now you can actually introduce anti-correlations. Okay? And in fact, you basically force, if these are the histograms of your correlation values, which have a positive bias, after global signal regression, you basically shift them all to be approximately 0 mean. Okay? And so the whole argument was simply that that these anti-correlations were not true and that they were simply an artifact. And so this paper was published like 10 years ago, and the controversy is actually still not resolved, even though you would think it'd be resolved because it's a really simple process. But I think one of the reasons in thinking about why it's not resolved is that if you think about it, even though it's simple, what we're doing is we're taking, say, 50,000 voxel time series, averaging them to come up with one mean time series, now regressing that out of 50,000 time series individually, and then trying to understand what's left over. And it's just that even though each step is very simple, you're still in a fairly high dimensional space. And so you can come up with a lot of theories of what's true or what's not true. And so a lot of the controversy has just been circular arguments going back and forth. So we decided to, with another graduate student, take another look at it that we thought was a little simpler way of thinking about it. And it's somewhat motivated by this quote by Yogi Berra. First of all, who knows who Yogi Berra is? Okay. So I'll have to explain who Yogi Berra is.
So Yogi Berra was a very famous catcher and baseball player, American baseball player, for the New York Yankees. So he was originally a catcher, and then later on, I think he was the coach, right? Right? Okay. And so he's really known for these, and he's also known as sort of in jest, like maybe somewhat true, like a great American philosopher, right? So he has these witty sayings, and this is one of them, which is, you can observe a lot by watching, okay? So the way I take this, and I think what I always stress for people, students especially in fMRI, is you just really have to look at your data. And the more raw form you look at the data, sometimes you'll see stuff that other people can't see because they've never actually looked at data. They just run it through a pipeline, and this is an example of what you can get if you actually look at data. So here, what I'm plotting here is this is just a single slice, but I'm showing it, this is time point per time point per time point, okay? So this is from time point 60 to time point 104. And what you see is there's a lot of things changing, right? And what you notice actually is sometimes the brain is really blue, which means it's very negative compared to its mean. So it's a negative global bold signal. Sometimes it's really homogeneously red, and sometimes it's in between. And so what these bars show are the global signal. So when the global signal is low, you expect things to be very homogeneous in the negative direction. When it's high, it's homogeneous in the other direction. And the really interesting thing, though, is when it is sort of low, you actually start seeing these interesting patterns. And this pattern here, it's sort of small, but we'll have bigger pictures later, is essentially you're seeing the default mode network and the task positive network just in a single time point, okay? And so the idea was that what we thought is, well, maybe global signal regression is just preserving these time points and getting rid of those other time points, or downweighting their contributions. And that's a much simpler way of looking at the data, right? Because you're just simply, each time point you can simply scale it and downweight it according to whether you think it's important or not. This slide down here below is just showing sort of global similar thing in a mouse model, showing you see similar effects where there are just these periods where the brain just lights up or becomes either very positive or very negative in a different animal model. Okay? So any questions up to now? Okay. So here's sort of the main thing. So you can take data, right, like this, and if you apply global signal regression, this is the data you're left with. And you'll notice that where this global signal is low, the data is fairly untouched by global signal regression, and where the data is high, it's fairly downweighted. And you can actually quantify that with what we call a GSR ratio, which is sort of the average downweighting factor. So it's highly downweighted here, here, and not so much here. Okay, so you can replace this regression with a very simple downweighting process. And you can use what we call ratio weighting, where you can actually use this ratio to do the downweighting, or you can take it to the limit, where you just zero things out if they're above a certain ratio and keep it multiply by 1 when they're above. Okay? And this is nice because now all of a sudden you have a different way of looking at what global signal regression might be doing. So just to summarize, this is the global signal. This is the ratio, and you sort of see when the global signal is high, we're really downweighting things. So we're almost multiplying by 0. And when the global signal is low, basically global signal regression leaves the data alone. All right? And then downweighting might be approximating with this linear weighting or with this hard threshold, which would be censoring. Okay? So I'll probably skip that slide.
Tom, can I just ask?
Yeah.
So I think regression is subtracting something out, but weighting is multiplicative. Can you help me a little bit more?
Yeah, it's an empirical observation, which is saying if you actually, it's actually related to leverage, okay, in the sense that if you have high points, those are going to drive your regression value the most, and that's what's going to get reduced the most. So if you can think of it that way, it's saying that when you have this things, so it's essentially, it's basically talking about, I can't pronounce the word, it's basically a temporal heterostochasticity, right? And so basically applying global signal regression isn't the right thing to do if you're looking at the data from that point of view. And so it's sort of, in retrospect, it's obvious that it's doing this, right? And it has to do this, So the nice thing is here, if you have uncorrected maps, so now these are different subjects, different runs. Global signal regression looks like this. Multiplying it by linear weighting looks like this, and censoring looks like that. Okay? And so they're not identical, but the major action of global signal regression is very well represented by this approximation. And it is because it's sort of approximating these leverage effects. Okay? With censoring, you can obviously vary different time points, but what I find amazing is, for example, here we're censoring out 87 or 86% of the data. We're only keeping 14% of the data, and you sort of see the map here is almost identical to what you get with global signal regression. Okay, so what it means is that if you do global signal regression, you're throwing away like over half your data. So you spend all this money to acquire data, you go global signal regression, you're essentially throwing away at least half your time points in some subjects. Okay.
And Tom, when you say it's large, you're just saying away from the mean or whatever?
Yeah, so if you have, right, so basically, so here's an example here. So here's the red is the global signal, right? So the blue is the global signal. The red is those time points we're retaining with this censoring of like 50%, or I think somewhere around, 0.5 is the censoring threshold. So yeah, it's basically just with respect to the mean. So it's pretty simple, but it's just a consequence of what regression does. Okay. So the nice thing, though, is that if you do censoring, right, for those data you kept, right, if there are anti-correlations in that data, you can argue that it's not a mathematical artifact. It was just there in the data, especially if you have a long stream of data. And you can find this if you look at data. There'll be long streams of data, 10 seconds, 20 seconds, where the global signal is very low, and you see the anti-correlations perfectly in that, and you don't have to process the data at all. Okay, so it suggests that these arguments that it's simply a mathematical artifact just aren't true. That really is something inherent in the brain, and also the fact that you can see it in a single time point is also, I think, evidence for that. So what it means is, anytime we do global signal regression, we're essentially decomposing our correlation map into some correlation map where there's low global signal and high global signal. Is essentially a temporal partitioning that you can sort of think of that as one way of doing it. And here's one thing I think is also adds to the fact that it suggests that this view of doing it allows you to think about that these anti-correlations are real, which is this is the traditional map you would obtain after global signal regression. Okay, so you see the nice default mode network and the anti-correlations. This map here was obtained after censoring, and it's simply the average of the images kept. It's not a correlation map. It's just I kept all the signals from the censored, the non-censored signals, and I just averaged them together. And so this is the very minimal processing, and yet you still see what's striking is basically all the information that we think is like correlation is actually just reflected in these mean signals. Okay, and so I think it suggests that even though everyone does correlations and it's sort of easy to do that, it may not be the right way of looking at the data. I mean, you might need to think about things going through different states and what they're visiting and that we might need to consider this alternative approach. This is very related to some work done by Xiao Liu and Jeff Dunn's group, where they've, this thing called CAPS, or coactivation patterns, where I think they were really the first to show that there are just the information. So this is a single frame, this is a single run, this is 247 runs, that this time frame average that they obtained basically can capture what's in the correlation map. Okay? So the idea is that it appears that the brain is just visiting these states that are really driving the correlation maps. And if you just look at those states, that might be a better way of looking at the brain. All right? So that's sort of a little bit about global signal regression. So the next thing I'm going to do is I'm going to talk about, well, what is the global signal? Okay? And some early studies, this was out of the NIH group, Scholvink and others, David Leopold's lab, where they stuck an electrode into I think primate, and they correlated the signal from one electrode with the fMRI over the whole brain, and they obtained these widespread correlations, okay, and so indicating that there was something electrical to do with that. This is from Zhongming Liu at Purdue, who looked at correlating different measures of broadband power, I believe, from EEG versus fMRI. And you once again see these very high correlations all over the brain. And this work is by Xiao Liu, who's now at Penn State. What he did was he took the HCP data. Actually, this was actually from primates. What he found was he could look at all the time points where there's high global signal. So these red things are a high global signal. And he had electrophysiological measures. And what he found out was that there was a stereotypical sort of EEG signature that happened every time before there was a global signal peak. Okay, so that there really was something preceding these changes in global signal. And then by looking at the HCP data, he was able to sort of locate it to, looks like this is nucleus basalis areas, some midline midbrain structures. So yeah, so nucleus basalis here, and arguing that there were these signals, some signal fluctuations in these brain stem structures, these deep brain structures seem to maybe give a clue as to when the global signal was going to be changing. Right. So I'm going to talk a little bit about some work we did in this area. And once again, we're going to go back to, these are data without global signal. This is with global signal regression. And here, we're not doing global signal regression, but we're giving the subjects 200 milligrams of caffeine again. And once we see that basically, although not exactly the same, it's doing something very similar to global signal regression. And so this pharmacological operation can also, it turns out, reduce the global signal. So this is the global signal amplitude, which is just the standard deviation of the signal. This is before caffeine versus sort of after caffeine versus before caffeine. You can sort of see there's a big reduction. Everything is below the line of unity. Okay. So this got us thinking and started a set of experiments where we use simultaneous EEG fMRI. The problem with the EEG fMRI, for anyone who's done it, is this is what the data look like. The EEG data look terrible, so you have to get rid of the gradient artifacts, then get rid of the cardioballistogram artifacts. And the data are still pretty bad, but at least you can work with them. Okay. What we'll use for our measure of vigilance is something that we sort of came up with after looking at the data, but then we found out it's actually a standard measure. So we called it vigilance. In the literature, it's actually called the alpha slow wave index. And it's simply the ratio of the alpha power over the delta and theta power. Okay? So there's other ways, obviously, of characterizing vigilance, but from a resting state point of view, this is a pretty good one. And so let's just walk through some of the data. So this is sort of another interesting I think from a point of view of how science gets done, another interesting story, which was, we looked at this data. This was a really intensive study. We took 10 subjects, we scanned them, 4 sessions, one with caffeine, and out, pre-post without caffeine, EEG fMRI, pre-post control with EEG fMRI. Then we also took them and did MEG, pre-post, and with control. So 4 really rigorous things. So we actually only got an N of 10 by the time we were done. And what happened was when we actually looked at the group differences, they weren't as big as we thought they would be. And we were sort of, had the usual depression for a few months while we're thinking, nothing's coming out of this data. And then we realized that it was actually the intersubjective variability that was the interesting thing about this data. Okay, that actually subjects responded quite differently to caffeine, and that we could use that to learn something about what was going on. So this is one subject who you could call caffeine resistant. Basically, this is that subject's fMRI maps, and these are the EEG spectra before and after caffeine. So very little difference. Here's the subject where caffeine makes a big difference. So here, they're relatively drowsy, okay, very high delta power, very low alpha power. The correlation map shows a lot of sort of signature of high global signal. And then after caffeine, alpha goes up, delta goes down, much less global signal artifact here. And so there's a big difference here. And so what you can do is you can now look at the change on a subject-by-subject basis. And so for every subject, you can look at the change in vigilance, and you can look at the change in global signal amplitude. And what you find out is it's a strikingly strong relationship. So this subject here, a big increase in vigilance, a large decrease in global signal amplitude. This subject here, almost no difference in either one. And if you look at all 10 subjects, they fit very well along this line. The other thing you can do is you can use eyes opening and eyes closed. So I forgot to mention we also did eyes open and eyes closed in this study. And it turns out that for some subjects, eyes open and eyes closed causes almost no change. In other subjects, you go from vigilant to non-vigilant. So you can play the same game. You can plot it, and you see the same relationship here. So delta change in global signaling amplitude versus the change in vigilance. But I think what's even more striking is you can take the eyes open, eyes closed data and plot it with the caffeine data, and it seems like they're following the same relationship, where this is, however you change the vigilance and how it seems to give the similar increase or decrease in global signaling amplitude. Okay. You can also look across subjects, and so here the relationship is not quite as strong, but certainly for subjects that are fairly vigilant, they have low global signaling amplitude. And then as they become less vigilant, the global signaling amplitude starts increasing. And then when they're sleepy, very high delta power, very high global signal. Okay. So I think this was our, whoops. I think I stepped on something. Yeah. Switch is off?
Okay. Have we done this one yet? Yeah, I'll pass it around and people.
Okay, great. Sorry about that. So the bottom line is I think that anytime we're dealing with global signal, it's not simply an artifact. There is a lot of evidence now that relates it to vigilance states, and I think that vigilance states is something that's fairly well ignored in most fMRI studies, but I think we need to think about it a bit more because it is highly correlated with a lot of different diseases. And I think having this in mind affects the interpretation of studies. So for example, this is a study from the Yale group published in PNAS where they observed higher global signal in schizophrenia versus the control group. And there's a very sort of model-based explanation for what this might say about the brain, but sort of a alternative explanation is simply that it's known that people with schizophrenia tend to have less vigilance than healthy controls. And so unless that's really controlled for, it's sort of difficult to understand what this data actually means. Okay? So I think this is something, whether you do global signal regression or not, I'm not going to say whether you should do it or not, but I just think it's important to keep in mind the information it might carry. So far we've looked at sort of overall global signal power. We can also look at, for example, on a time point by time point basis, and what we find there is that, here in red is the global signal, and here in blue is the vigilance time course, and we find there are significant correlations, and it turns out that to first order, whenever the vigilance is low, the brain seems to have more oxygenation in it. Okay, so the bold signal is higher overall. We think this might be related to adenosine effects. So and the clue there is that caffeine is an adenosine antagonist, and so one possibility is whether these are adenosine-related fluctuations, but I don't have a sense of how quickly those can change. And then with high vigilance or relatively high vigilance, then it tends to be that the bold signal just tends to be lower overall. Okay. So what that means is if you go back to sort of our previous view of, if you did global signal censoring, you would retain, if you retain the time points with the low global signal magnitude, that's where the anti-correlations are. And what you're doing is you're really censoring out the points that are, as compared to the mean, very high vigilance or low vigilance, right? So you're sort of ignoring when the brain is going through these sort of extreme values of vigilance, and you're just keeping the values in between where the brain is sort of in this, this mean state appears to be immune from those vigilance fluctuations. So it may be that one thing we're doing in global signal regression is reducing these vigilance fluctuations in the data, which might be a good thing or might be a bad thing, depending on your point of view. Okay? So depending on what your experiment is doing, that might be good. And certainly it fits in the sense that the reason why global signal regression works so well is probably because there's going to be lots of variation in vigilance across your scans and subjects, And this is one way of reducing that variability. This is some nice work done by Katie Chang out of Jeff Dunn's group at NIH. This is the same primate data as the Scholving paper, and what she noticed is that these are monkeys like in a dark room for like an hour, and they're basically struggling to stay awake. And so what she did was interesting. She just basically looked at how open their eyes were, and so these black graphs are sort of a measure of eye open versus eye closed for 4 different monkeys. And what she could do is she could then correlate this time series with the fMRI data and come up with a template. And then she could use that template on some other data to predict what the arousal might be. And as you can sort of see here in the red, this is the predicted arousal. This is the actual arousal. And it seems to be a very nice correlation here. Okay? And so, so it seems to be there is some ability from the fMRI data alone to predict vigilance fluctuations, which might be useful for some studies. Now, the sort of the not so promising news is then we worked with Katie and we applied this to our fMRI data, where we had the EG data and the fMRI data. And what we found was that, so this is the template you get before caffeine, so a very strong template, very similar to the template she's getting, although this is monkey, this is human. But both templates typically involve thalamus, and it's anti-correlated with visual areas. But on caffeine, the fluctuations are much less, okay, in the sense that we know caffeine reduces the global signal, and so it reduces these vigilance fluctuations. And therefore, the ability to predict vigilance fluctuations across scans may be limited by the fact that there is this variability in how much it fluctuates. So what we can predict may be limited, but we have found that we can predict sort of the standard deviation of the fluctuations, and that might be useful to know. Okay. So that's all I'm going to talk to you about with global signal and vigilance. So if there's any questions, let me know. All right. So now we're going to move on to, so the rest of the talk is mostly going to be talking about some things we've been doing in the last couple years on nuisance regression. And just to remind you, dynamic functional connectivity, we're doing a sliding window calculation, and whether you believe in this method or not is sort of doesn't matter. It's what people are doing, and so we're going to take a look at it and point out some issues that it might have. So let me just walk through one way that people analyze this data, and then actually I will still talk about arousal because it's related to this. So here what they've taken group ICA, they get these different subject time courses, they do windowed correlation on the ICA time courses, they aggregate the data, these correlation matrices over subjects, and they do k-means clustering. Okay? So they're arguing that, for example, in this study, they're arguing that there's 5 different states, and each state is represented by a correlation matrix. And in this study, they found that healthy controls spend most of their time in these 2 states, whereas schizophrenics spend most of the time in those 2 states. Okay? So once again, a very nice, quote, biomarker type study showing that there is a difference between 2 populations. But this, so that was a 2014 paper. This is a recent paper, 2017, where they did the same thing. So here are the different clusters that they have. So this is cluster 1, cluster 2, cluster 3, cluster 4. And so these are the bold time series. And what they also had is they had EEG. So when each cluster is active, they could also tell whether the person was awake or N1 or N2 or N3 sleep. Okay? And what they found was basically whether you use the EEG to do your clustering in terms of sleep states or whether you do this k-means clustering, you basically obtain the same things. And so these states that people are going through may just be different states of wakefulness. And that really sort of fits with the schizophrenia data where probably if the schizophrenia subjects were in N2, N3 more or closer to wake or N1 for the control group, that's something that just doing a blind analysis you wouldn't necessarily know. Okay? And for resting state, it is a problem because this is a paper looking at the probability of sleep, and this is using fMRI and EEG. And basically after about, after about 2 or 3 minutes, most subjects are asleep or falling asleep very rapidly, okay? So I think, for example, any of you looking at the Human Connectome project data, those are 15-minute long runs, resting state runs. And anyone who's been in a scanner and done these things, you know that that's really not resting. That's not very restful. To stay awake with your eyes open, fixated for 15 minutes, it's like the worst task possible. And so I think these are, so as you're analyzing the HCP data, you have to realize what you're really measuring is people struggling to stay awake, okay? And so I think these vigilance and arousal effects are probably highly prevalent in the HCP data. With newer projects like the ABCD, I think they're shorter scans, but even so, you know, if you're on the second or third of a scan of a 6 minutes, you know, if you've done 3 6-minute scans, you're already pretty tired by the third scan, and there's going to be clear arousal issues. Okay. So one thing we did, we were interested in looking at, you know, how global signal regression affects dynamic functional cognitivity. But what we realized later on in doing that is actually it's a much more prevalent problem. So here's, I'm going to outline what we did. So here we did sliding window correlation. So for every window, you get the sliding window correlation between two regions. And then what you can do is you take any nuisance term. You could take motion, you could take white matter or CSF or global signal, and you can simply, within each window, there's different ways of doing this. For this study, we just took the L2 norm, or so essentially the standard deviation of the time course in that window, and you can end up with what we call the nuisance norm, and you can correlate the two. Okay, so you're just arguing over windows, how related is your correlation estimate to your nuisance norm? And if they're related, then you have to question what is your functional cognitivity estimate telling you? And the bad news is they're highly related. Okay, so that's the bad news. There's worse news too, but first let's talk about the bad news. The bad news is, so here in red, or sort of blue, is your functional cognitivity estimate, and then in black is the nuisance norm. And obviously these are picked to really highlight it. It's not always this case, but like 30% of the scans we looked at have this problem. Okay, and so that's the bad news. The worst news is you'd think, okay, no problem. I'm just going to regress these effects out of my data using the typical general linear model. And so if you do that, you end up with the red curves, okay? And so regression actually doesn't help. And so that's sort of fairly surprising, counterintuitive, but I'll sort of explain sort of why that might be true, okay? One thing that's sort of interesting is what we did was we said, well, you know, why is this happening? Is this because, you know, it sort of makes sense if the nuisance regressor is really highly correlated with your time series, that would make sense that the nuisance norm might be highly correlated with these windowed estimates. But what we found was, even if you have a very low correlation, let's say a correlation of 0.1 here, between the essentially the time series and the nuisance regressors, you're still seeing points. So even these time points, sometimes the correlation with the functional cognitivity and the nuisance norm is as high as 0.8. So even when you have very little relationship between your measured time series and your nuisance regressor, the correlation over windows can be very strong still, which was also sort of counterintuitive to us. Okay? So let me just show you a toy model of why that might be plausible. So this is something where we're going to represent time series with vectors, okay? And so here, a very simple model, let's assume y1 and y2 have the same correlation throughout time. So your null hypothesis is that there's no change in correlation. That's the true underlying data. We never see that, but in this model we can posit that that's true. Let's add a nuisance term in plane, and that nuisance term varies in amplitude, right? So its norm is varying, and therefore that's going to change this angle, and the correlation is just the cosine of that angle. So the correlation is going to go up and down with the nuisance norm. Okay? So at least it's plausible that a nuisance norm would change the correlation values. Okay? So you have no change in correlations. In the green, you have this nuisance term. It can drive all these changes in functional cognitivity. So, but then how would you get, in this case, you could argue that, well, maybe you know, the angle between the nuisance norm and each of these vectors, I mean, you could imagine it could be smaller, and or you know, shouldn't this be able to, shouldn't you be able to take this out somehow? Can't you regress that out? And the problem is that, you know, this is a 2D picture, so it's very simple, but in reality what we're faced with is more like this 3D picture where these are your observed data here, Okay, and these data are identical to the what I just showed you in 2D. Okay, but now you can posit that the nuisance norm is made of two components. One is an in-plane component that is in plane, and there's this orthogonal component that's outside. And if those two things are related, then these guys can be highly orthogonal to your data of interest, but because their in-plane component is varying in relation to that norm, they're still going to drive your correlations. And so that's exactly what we find is essentially here you have the nuisance norm going up and down. Okay, here you have the before regression, and then after regression, there's almost no change because of how we've set up the model. And obviously, this is an explanatory model, so we've made it so it looks like that. Okay, so I'm not going to go into the details so much, but just sort of point that out as the underlying theory is we're now able. What we did was we went through and we derived some bounds on delta F DFC is basically the FC after regression minus the FC before regression. Okay, and it turns out that that is bounded by this equation here, which is related to how much of the this n naught squared over n squared is how much of the energy of the nuisance norm is out of plane. All right, and you end up with these fairly tight bounds. Okay, and most of the time we're operating in this region, and so basically because of these theoretical bounds, whenever we do nuisance regression, we're very limited in how much we can actually affect this relationship. And then what happens is even if you sort of go down here where there's very small orthogonal fraction, it seems that most data follows this bound pretty carefully and hugs that barrier. And so you're still left with this very narrow band of here there's a big change, but because it's almost an offset, you still end up with this correlation that lasts. Okay? So the main thing is that it's counterintuitive, but there is sort of, you can actually explain it all just with some equations. And it's something that I think we need to keep in mind for dynamic functional connectivity and it's something that people really haven't looked at before, right, in terms of the FC measures over time and then how related to the nuisance norm. And so I think people probably still just need to start looking at that, because if all your dynamic functional connectivity measurements can be explained by these nuisance norms, then it sort of begs the question of what are you actually concluding. The last topic I'm going to talk about is taking the same theory to static FC, where instead of windows, you have scan 1, scan 2, scan 6, scan K, right? Those are just bigger windows, right? Different subjects, different scans. The same principle applies. You can take the nuisance norm from each scan, and the FC estimate from each scan. You actually get very similar relationships. Okay? So this is just showing that it holds for different regressors, and that even if you do nuisance regression, that relationship still holds. And so even studies that are looking at cross scans are going to have this issue. And one way we can look at that is we can, for every scan, we can compute, sort of say this is the seed signal. We can compute the correlation matrix here, which you can represent as a 1D vector. And then you can sort of stack all these 1D vectors across time, across subjects and scans. And so for every voxel, okay, you have this connectivity varying in time, or varying across scans. And you can correlate it with how the nuisance norm varies across scans. And you can end up with what we call a contamination map. So for every voxel, how contaminated is your data, are your functional connectivity estimates across your study by this nuisance norm fluctuation. And so for certain regressors, for example, head motion regressors, you're highly contaminated. And even after you do regression, you're still highly contaminated. Okay? Now, this isn't totally surprising. It's known that head motion regressors aren't very effective. And so maybe you want to up the game a little bit and go to like global signal regression. And so let's take a look at that. This is just a slide showing that the same bounds apply. But let's spend a little more time on this one. So this is global signal regression. And so here, before you do if you don't do any global signal regression, your functional connectivity estimates over your study will be highly correlated with the global signal norm. Okay? So you say, great, I'm going to regress global signal regression out of every scan. And what it does is it actually introduces this really interesting picture here, where now all of a sudden, sometimes you still have a very high positive correlation, but now you've introduced these anti-correlations. And these are very different from the anti-correlations we talked about before, because these are anti-correlations across scans, okay, not within a single scan, all right? So what it's saying is that depending on what brain region you are, global signal regression can have a very different effect on how you might interpret variability in functional cognitivity across your scan, okay? Now, it turns out the reason that they are related to the default, sort of the anti-correlations we saw before, is because the correlations, they depend on the average correlation with the voxel of interest, all right? I'm not going to go into the details, but let me just give you a quick summary of that. So in this zone where there's anti-correlation, so this guy here, it tends to be those areas. So these are the functional cognitivity values across scans versus the global signal norm. So you see this positive correlation, but you sort of see the slope is relatively weaker as compared to this, where the slope is much steeper. And so this is showing a much stronger relationship between the functional cognitive measures and the global signal norm. In both cases, because of this theoretical bounds, the change, so we're going to take the blue plus the black to get the red. This is all that global signal regression can do. That's the most change it can implement. It's because it starts actually hitting this barrier here, and it can't change the correlation values anymore. So what happens is this slope is essentially the same between regions, but adding this to this gives you this negative relationship, whereas adding this to this, you still get a slightly positive relationship. And this is totally dependent on whether you're highly correlated with the PCC seed or less correlated, and so that's why you end up with similar maps. Okay? So the take-home message here is that I think even though nuisance regression, we sort of assume that it's working for us, I think we may need to be a little more careful, and if we look at data in a slightly different way, we may find out it's actually still in our data. We haven't taken it out. Okay? So to summarize, I think it's good to keep in mind that global activity can make up a significant portion of the resting state signal, and it appears to be related at least somewhat to fluctuations in arousal and vigilance. However, I think we need to understand this a little more. And keep in mind that it's highly variable across scans and subjects. Okay. Regardless, whether you do global signal regression or not, I think it's good to keep in mind that arousal and vigilance variations are going to have a major effect on your functional cognitive measures. And I think this suggests the need for additional measures of the subject's state and possibly new analysis approaches. So I think one thing is, for example, especially if you're doing eyes open fixation, I think having a video camera of the eye should be the standard process, because you will just see the eyelid fluctuating quite a lot. And it's actually quite amazing. You can just sort of see what their level is with just this camera. Okay? And I think this is going to be especially important for studies where you sort of already go in knowing that there's going to be differences. So for example, with development or autism or schizophrenia, I mean, there's already evidence suggesting that it's going to vary. And so if you ignore this, then you're sort of ignoring a large part of the variability. And finally, I think for the most part, most of us use nuisance regression, and we assume that it's getting rid of all of the effects. But I think it's good to keep in mind that, especially for linear regression-based approaches, there are these limitations. And so whether or not there can be better methods developed in the future, I think that's an open question. And then, and so that's what that says. And I'll leave you with this slide, which is, I think, something I've been thinking about more recently is just the fact that we sort of make this arbitrary delineation in fMRI between signal and noise, right? Motion is noise, respiratory compounds are noise. But I think there's growing evidence. I mean, obviously, we know the brain controls pretty much your heart, your respiration. There's always this feedback loop, right? Motion, you move, you react to the motion. And so I think that there is going to be a need, I think, for maybe taking a more holistic view of how to analyze data. And I don't know what the solution is, but the more I look at sort of this field, I just think this simple dividing line is really not, at least in my mind, the right way to go. Okay. So with that, I'd like to thank you for attention. Oh, sorry. I didn't realize, keep forgetting that you guys don't see what I see. So has this been the case for the whole talk? Have I always been a slide behind? No, okay, just these last few, right? Okay, thanks, good. Phew, all right. Okay, so this was the slide here, which is basically, the brain is controlling the part, the respiration. CO2 modulation, we know modulates cerebral blood flow. We know this perturbs the magnetic field. We know there's motor control, there's head motion. All these things go into fMRI signal. And typically, we just draw a circle around the brain, and everything else is noise. But I think it's worth thinking about whether we need to draw a bigger circle. Okay. So with that, I'd like to thank you for your attention, thank people who did a lot of this work and the funding sources, and happy to answer any additional questions.
Questions from the audience?
Okay.
Quick question.
Sure.
So, one of the methods you said was to actually look at their eyes, some kind of eye tracker camera, something like that. Has anybody ever done anything like a subjective report? Like how well do you think you were staying awake during this whole thing, or actually doing what we told you to do during the resting state? And does that also correlate with these?
Yeah, that's a good question. I think there is a resting state in questionnaire. Has anyone used that? I know one of the groups, I think there's like an Amsterdam inventory that you're supposed to do afterwards. And I think they've done some, I think there is a paper out there where they've correlated that with some of their findings. So that's, but I don't remember the details of that. I mean, I think that's certainly good to do, but obviously it's, it's going to be highly dependent on their memory. So I think if there's simple things we can do that actually get at it, then that's going to be preferable. I mean, to a large extent, I mean, you can use things like heart rate variability potentially. We talked about pulse amplitude. So I think there's just a lot of other measures that if we take into account things more holistically, might give us a better way of looking at the data. Yes?
So you talked about this,
mostly in terms of resting state.
Yeah.
Because that's where a lot of the controversy is. But
Right.
presumably the same thing will be going on, in task-based fMRI too, because certainly you have a long run people pay more or less attention. Like I'm thinking. So could you actually use this, like, so we were talking about predicting behavior before.
Yeah.
In this case it'd be post-addicting, but let's say like, when people are doing like the SART, sustained attention response tasks, like can you use this to predict like when they start speeding up or making an error or something along those lines?
Yeah, I mean, we haven't looked at that, but I think it seems like it's reasonable to do that, and certainly there have been some studies looking at, certainly in the EEG literature, depending on what phase of the alpha cycle you're in, it has a big effect. And I think there have been some studies looking at the fMRI signal in the default mode network, and as a proxy for where your attention is. So I think that's definitely doable. So yeah.
Okay. So just one last thing. So you ever looked at an ASL signal with the Vigilance and see how they correlate? See if the blood flow's going up and down? I guess it's like really noisy, but.
We haven't looked at it dynamically. I mean, the only ASL we've done is with the caffeine. We did measure the blood flow, and for those of you who don't know, caffeine reduces your blood flow by about 20 or 30%. So,
which is the natural vigilance measure that you were doing with the EEG. Is that
yeah,
with the flow,
I don't. I mean, I imagine so that's a good question. I mean, I think it would be, and we've talked about, it'd be nice to look at sort of overall CBF levels and how they're affected, but I think it's confounded by, we know, for example, with caffeine, there's a definitely overall decrease in blood flow, but that's related with, that's less global signal. And so the thing there is, typically, if you just went with bold modeling, you would think if you decrease the blood flow, you have less deoxyhemoglobin in the baseline state, so it should increase the bold fluctuations. And certainly that might be an explanation for it. But I think the actual explanation for it was more there's changes in arousal. And if it is changes in arousal, I expect the CBF to follow that. I think there is some evidence maybe from the NIH group looking at that with sleep. And so I do think that even, any sleep studies that look at blood flow either with ASL or with laser Doppler, I think would be interesting to look at. Yes?
Have you done ADHDs on and off medication?
No, but I was talking to Chandra about
Okay.
it a little bit, that he has a lot of data that seems like that might be interesting to look at.
Excellent timing. I guess
Okay.
if there are no more questions, we'll just thank our speaker.
Great, okay, thank you.